Hello and a very warm welcome to our webinar today, which is about advanced BB-on-chip models for brain cancer research. Also, this will be our very first panel discussion, will be after the scientific presentation. And it will be about current bottlenecks in brain cancer research and the importance of advanced preclinical systems for translational success. Before I move into introducing our speakers today, I would like to go through a bit of housekeeping. So if you look on the left-hand side, you find a control panel. Very important, there is a chat and Q&A function. So if you have any general inquiries, I would ask you to use the chat function. Should you have, or hopefully you will, have questions to our speakers, you can use the Q&A function. So I'd very much like you to ask to use this, because this will really help us nicely prioritize the questions that are coming in by the audience. And lastly, we will record this webinar, so you will get the recording after it's concluded, so that you can rewatch anything that you thought was interesting or share it with your colleagues. So now, without further ado, I'd like to introduce our speakers today. So joining for our panel, we have Dr. Jens Hoffmann, Dr. Martin Raasch, and Dr. Thomas Sommermann. Jens is a CEO of Experimental Pharmacology and Oncology Berlin Buch GmbH, and a pharmacist with a diploma in pharmacy, experimental pharmacology, and toxicology. He earned his PhD from the Humboldt University Berlin and held research positions at MDC, DKFZ, Schering AG, and the University of Pittsburgh. He's led oncology research and translational drug development for more than 2 decades, with over 90 peer-reviewed publications and more than 30 patents. Quite impressive, I might say. Then we have Martin. He's co-CEO and co-founder of Dynamic 42, and he's been working with on-chip cell culture applications since 2010 as part of his master thesis at Leipzig University, and intensified his research during his PhD in cell biology in the group of Dr. Alexander Mosig at Jena University Hospital. Since 2021, he is also a board member of the European Organ on Chip Society, where he's leading the working group on science and technology implementation. Then Tom completed his PhD at Harvard, where he worked on lymphomas associated with Epstein-Barr virus, and he continued this work later on at the MDC in Berlin. At Dynamic 42, Tom is now heading the cancer team, and he's working on the development of our cancer spheroid-on-chip models. So then, after a bit of introduction by Jens and Martin, Amélie Paillereau and Michelle Zimmer will present their scientific data on the BBB models. Amélie studied biotechnology in Jena, where she completed her bachelor's degree in 2019 and her master's in pharmaceutical biotechnology in 2024. Since April 2024, she's been a PhD student in the cancer team at Dynamic 42, and she's researching the human blood-brain barrier on chip. And then last but not least, we've got Michelle. She studied biotechnology at Technical University Berlin, where she completed her bachelor's degree in 2023 and her master's in 2025. She joined IPU as a student assistant 2 years ago and began her PhD there in October, and is now continuing her research on blood-brain barrier models and glioblastoma. And with this, I'd like to hand over to Jens.
Hello and good afternoon from Berlin. Today we have a presentation and webinar between 2 companies, 2 research companies, and we will shortly introduce our companies, and I will start with EPO, and EPO stands for Experimental Pharmacology and Oncology. This already tells you what we are doing. So we perform preclinical oncology research. We are meanwhile since 30 years in business, located in Berlin Buch. And we perform preclinical studies to support drug development projects in oncology. We have supported research for more than 20 compounds, which meanwhile have been approved by the FDA. We have performed more than 20,000 studies, more than 600 publications, and altogether our research available, and this work is done here in Berlin by approximately 50 employees. And we work worldwide and have ALAC certification, and everything in the building you can see here on the picture. And if we go to the next slide, I can tell you a little bit what we are doing. So we have this large panel of tumor models. We perform drug testing. We have humanized models for immuno-oncology research, but we can also support target validation with some more than 1,000 tumor models. We have technologies here in-house spanning from live cell imaging, flow cytometry, high-resolution 3D imaging, live cell imaging, and we can also perform experiments with radiation and with radionuclides. And we have a histopathology lab, bioinformatics and biostatistics. We can also perform studies for toxicology, PK, PD research. And with this, I would like to hand over to Martin for also a very short introduction of Dynamic 42.
Thank you, Jens, and a warm welcome to everyone in the webinar this afternoon. Dynamic 42 is an organ-on-chip company. We are situated in Jena, in central Germany. We put here the chart because I guess Berlin, everyone knows. But Jena is a way smaller town, but has a very large life science community. And we started with our research and work at the local university hospital and spun out several years ago. And on the next slide, thank you. I'll give you a brief overview what we are doing. Of course, our organ-on-chip platform that we developed is available as a product to end customers, especially in the academic space, but also in the pharma space. We offer a variety of different chip designs to address different specific research questions so that everyone can tailor the platform to their needs. We also provide services in the preclinical in vitro testing area, where we work mainly with larger pharma companies or also biotech companies together. But we provide also services in terms of developing new chip designs for special research questions. And last but not least, we strongly believe in training and education. And since organ-on-chip has been around for a while, but is still new to a lot of researchers, we provide training courses, hands-on training courses here in Jena in our site, but have also an online tool where people can learn with our platform to build their own organ-on-chip models. And I would like to hand over to Jens again, who will tell you a bit about the motivation behind our collaboration and about the project.
Yeah, thank you very much, Martin. If you are working in drug development, you know that the most critical path is to really find out how your research is translating then later to the humans. And if you would like to do this, you need models. And in drug research, you usually use models like it is shown in this pyramid. So at the beginning, you start with high throughput screening in 69 well or 348 well plates. So you can evaluate a lot of compounds. But then you have to address more specific questions. And here the lab-on-chip technology is extremely helpful meanwhile, because here you can really test compounds much more closer to the human situation. And this helps, this research, this technology really helps to select only very active compounds where you have a very high certainty that they will later work in patients. And with this, you can really reduce the number of animal experiments also on this side, so that really only very active and very promising compounds go in the next step in the xenograft model, especially in oncology. And with this all together, then you have really very safe and very promising compounds, which then can, in the last step, be used in human trials. And Martin again will show you a little bit more about these corresponding technologies.
Yeah, with this, what we try to build here, we really want to set up a nice toolbox where you have all advantages and combine different models so that in the end you create a very large network of data and have a high certainty to go into human clinical trials, for instance. So we're not talking about just 2D in vitro experiments anymore or single animal experiments. With this collaboration, what we also started, we really see the benefit that you can set up human models on chip. You can also set up these as a mixed model, so-called xenograft model on chip, where we have, for instance, animal components building the normal organism in a simple way, for instance, the blood vessel, and we bring in the tumor cells from the patient to do a xenograft model on chip. And in parallel, we can do this also in a animal model, in a mouse model, and have here the possibility to compare the data of all different approaches and get a very comprehensive picture before we go into human clinical trials. And this systematic cross comparison that you can do here is not just limited to one tumor. This can be done with different kind of tumors. And we have selected glioblastoma for a start. And with this, I would like to hand over to our two main actors here, Amélie and Michelle, to present the start of our project and what we have achieved so far. It is not completed yet, but both want to give you first impressions where the journey will go. Yeah. So the stage is yours, Amélie and Michelle.
Thank you very much, Martin. Building on this translational framework we just saw, I would like to introduce you to our field of application on the scientific context of our work, the blood-brain barrier modeling in glioblastoma research, as Martin already said. Glioblastoma is a highly aggressive primary brain tumor with a 5-year survival rate of only 5%, and it is characterized by rapid growth, strong cellular and molecular heterogeneity, as well as a high recurrence rate. Even though the blood-brain barrier, short BBB, remains a major obstacle in brain tumor treatment, glioblastoma is also associated with a partial BBB disruption, forming a heterogeneous blood-brain tumor barrier, which affects the already complex and immunosuppressive microenvironment. And one major problem in brain cancer drug development is that current in vitro and animal models do not fully capture the complex human BBB tumor interactions. And to move forward or towards more physically relevant in vitro models, we focus on patient-derived systems. At EPO, we currently maintain 40 established patient-derived xenograft glioblastoma models, short PDX models, which we use to generate corresponding cells or spheroids for in vitro applications, and receive the patient tumors tissue samples from the hospital, including the human cancer cells, tumor-associated cells, and other human cells. And this tumor material is then engrafted into mice. And after successful growth and passages of 2 to 3, the PDX models are used for in vivo efficacy studies under standard treatment conditions. And in parallel, the passages 2 to 3 tumors are used to establish the in vitro cells. Therefore, we dissociate the human tumors from the PDX models mechanically and enzymatically into single cell suspension and seed them on a extracellular matrix to ensure that we have a mouse-free cultivation. We perform a mouse cell depletion and selectively culture the human cancer cells using specific media conditions. And this whole process takes about 4 to 12 weeks in total. Our established cells show adherent growth with a brain tumor-like morphology and partial glia-like features, as you can see in the top images of 3 selected lines. And our models, most of our models also form 3-dimensional structures in a low adhesion plate. And to compare if our established cells and the original PDX tumors are similar, we performed RNA sequencing, revealing that highly similar gene expression patterns could be seen, and with similar up and down regulation of selected genes. So this indicates that our cell models closely recapitulate the characteristics of the original PDX models. However, even with robust glioblastoma cell models suitable, for example, high throughput screening in plate-based systems, a major in vitro limitation remains the selective barrier function of the BBB. So what is the BBB actually? It separates the blood flow from the central nervous system in the brain and is mainly formed by endothelial cells, which are supported on the brain-facing side by pericytes, a dense basal lamina, and astrocytic end feet. And the endothelial cells of brain capillaries are connected by very tight and dense cell-cell contacts and characterized by strong, tight, and adherence junctions. These structures alone already restrict the passage of large and hydrophile molecules into the brain bigger than 400 Dalton. So in addition, efflux pumps like P-glycoprotein, breast cancer resistance protein, or multi-drug resistance associated proteins actively transport molecules out of the brain. As a result, the brain is one of the most well-protected organs, which also means that many drugs are prevented from entering into brain tissue, and more specifically, more than 89% of small molecule drugs. And that is actually a big problem for drug development.
So even though the BBB plays a crucial role in the context of disease and more specifically in the context of brain tumors, human in vivo research on the BBB is still quite tricky. We can get an indirect indication on the BBB through contrast enhancement in MRI. However, there's still the need for more advanced models to enable more direct research on the BBB. Murine models already enable fairly direct research on the BBB. However, they cannot replicate the species-specific interaction between brain tumors and the human blood-brain barrier. Mouse brain endothelial cells differ, for example, from human brain endothelial cells in transporter protein expression of key transporters, such as PGP or ABC transporters, that affect drug permeability across the BBB, but also they differ in tight junction protein and enzyme expression. In addition, the mouse BBB is believed to be more permeable than the human BBB, which then again limits the translational reliability of preclinical findings. And finally, the neuroimmune environment in mice is less complex and different to that of humans, especially in the context of immunocompromised mice that are used for PDX. So two-dimensional models have in vitro long been a cornerstone of cancer research, as they are cost-effective, they are reproducible, and also easily manipulated platforms for studying tumor biology, doing drug screenings, and also looking at cellular responses. However, these 2D, even co-cultures, fail to recapitulate important characteristics such as the complex three-dimensional architecture of tissues and tumors. And especially when we are looking at a tissue with a barrier function, dynamic transport and efflux mechanisms cannot be replicated in a two-dimensional structure. So as a first step, three-dimensional co-cultures, such as tumor spheroids, are widely used as 3D models for the study of tumor biology, which already replicate key features of solid tumors. But then again, when we are looking at vascular tissues, where the blood flow is relevant, three-dimensional co-culture models, such as transwells, do not incorporate dynamic flow conditions, therefore limiting the physiological relevance of these models. To overcome these limitations, emerging tumor-on-chip platforms are recapitulating multicellular composition, dynamic flow conditions, and three-dimensional architectures of the blood-brain barrier, thereby offering a promising alternative. And this is where Dynamic 42 comes in again, as they are providing the biochip platform for this organ-on-chip research. How does the biochip from Dynamic 42 look like? So each chip, as you can see here, comprises 2 individual cell culture chambers that are each divided into multiple channels via porous membranes. In this model, we are using a 2-channel biochip, as you can see here, which has 2 ports for the manipulation of the top channel and 2 ports for the manipulation of the bottom channel. Zooming in one of these cell culture chambers, you can see the division into a top channel and the bottom channel via the membrane, as I mentioned before. We can seed in cells to the top channel that will grow on one side of the membrane, but we can also seed in cells through the bottom channel, turn over the chip to culture cells on the underside of the membrane. When growing endothelial cells, we can mimic the blood flow through perfusion of media via a pump, as we are showing here for the top channel. And this is then how it looks like. So the top channel is hooked up to tubing and a media reservoir, and all of this is connected to a pump on the inside of the incubator. The perfusion not only provides continuous nutrient and oxygen supply, but also induces shear stress on the endothelial cells, as we have in vivo in our blood vessels. But now, coming back to the blood-brain barrier and glioblastoma model that we want to build in this Dynamic 42 biochip. This is how this looks like. So you can see here again the division into a top channel and a bottom channel by the porous membrane. And to model the blood-brain barrier, we are seeding brain endothelial cells in the top channel. Underneath, as a monolayer for now, we are seeding glioblastoma cells in the bottom channel. And the top channel then mimics the blood side of the blood-brain barrier, while the blood channel represents the brain side. Perfusion is then applied to the top channel with the brain endothelial cells. To determine the impact of the perfusion or even of the glioblastoma cells on the blood-brain barrier, we chose 3 different readouts, such as immunofluorescence staining of the endothelial tissue, which we recover out of the biochip, but also permeability measurements via fluorescent labeled dextrans, or measurement of transendothelial, transepithelial electrical resistance. And what we are, of course, also doing is comparing the mouse and the human blood-brain barrier. What you can see here are IF images of mouse and human brain endothelial tissues that we have stained for tight junction proteins such as PKM1, Claudin-5, and Zonola occludens 1. Just by comparing the endothelial cells, we can already see clear difference between the 2 species. The cells differ in size and morphology. What I just want to briefly mention here is that the mouse and the human images in here have different scale bars. But even though you can still see the size difference of the brain endothelial cells between those 2 species. Also, the mouse endothelial cells arrange themselves in an elongated shape, while the morphology of the human brain endothelial cells is more similar to the morphology that we know, for example, of HUVECs. Also, in the response to perfusion, or more specifically the shear stress induced by the perfusion, the mouse endothelial cells could withstand higher perfusion rates, even up to 50 microliters per minute, while the human brain endothelial cell layer was already disrupted at higher perfusion rates. So for further experiments, we chose the conditions for each models with the most intact brain vascular tissue. For the mouse endothelial cells, tight junction protein, for example, was the best at 25 microliters per minute, which is why we went for this condition for further experiments, in which we did drug application against the glioblastoma blood side of the barrier. For the drugs, we chose 3 different drugs that had an inhibitory effect on our glioblastoma cells in previous cell plate experiments. And the results of these chip experiments, Michelle will tell you a little bit more about.
Yes, to qualitatively analyze the effects of the drug treatment on our endothelial monolayers, we assess the expression of adherence junctions and tight junction associated proteins, the cadherin and PKM1, and after treatment of 3 days. As already observed in the endothelial cell behavior, mouse and human cells also show distinct responses in this context. In human endothelial cells, drug 1 and 2 induced some off-target effects, whereas in the mouse endothelial monolayer remained largely intact. So only with treatment with drug-free, there were discontinuities in the junction integrity observed in the human cells, while in the mouse cells, this treatment led to a more pronounced detachment of the monolayer. And to quantitatively assess the drug efficacy on the glioblastoma cells and the BBB permeability, we evaluated the tumor cell viability after the 3 days of treatment across the species-specific endothelial barriers. And despite the off-target effects in human endothelial cells, which we saw in the previous slide, with drug 1 and drug 2, the components seem to not cross the barrier, as there was no inhibitory effect on the glioblastoma cell proliferation. And however, in the mouse endothelial barrier, which remained intact in drug 1 and 2, we could actually see that drug 1 showed strong inhibitory effect on the tumor cell proliferation, contrary to the human barrier system, indicating there was a barrier passage. In addition to its effect on the endothelial barrier, drug 3 had also an inhibitory effect on the glioblastoma cell viability in both systems. So that actually fits together. And to further improve the physiological relevance and reduce destabilization of the barrier by flow or components, we are currently expanding the system by incorporating additional key BBB components like parasites and astrocytes. And in our chipset app setup, the top perfused channel is again lined with endothelial cells, while a parasite monolayer is seated on the basal part of the membrane in the bottom channel, allowing direct contact with the endothelial cells. And in addition, astrocytes are also incorporated in the bottom channel. In this extended BBB model, we analyzed changes in barrier integrity using tear measurements. Transepithelial electrical resistance is a measure of barrier tightness, where higher values indicate a more restrictive and tighter barrier. And we observed improved barrier integrity over time in the presence of parasites, as well as in co-culture with both parasites and astrocytes, as you can see on the left diagram of the mouse model. In a permeability assay in the human model on the right side, using fluorescently labeled polymers, we could also observe a 50% reduction in permeability in co-culture with parasites compared to the endothelial monolayer alone, indicating that there's also an improved in barrier integrity. And we also expect that the barrier integrity in the human model will also be even better with astrocytes and parasites in co-cultivation, to be seen as exactly as in the tear in the mouse model, leading to a reduced permeability in co-cultivation with both. So in the future, the expanded BBB model will not only include a monolayer of tumors like we did in the experiment we showed you, but it will be further developed by integrating the patient-derived glioblastoma spheroids, which I was talking in the beginning about. And this aims to establish a stable in vitro platform that bridges the plate-based screenings and xenograft models, enabling studies in brain cancer, BBB function, and drug development. To summarize our presentation, we already observed differences in morphology, behavior, and treatment responses between mouse and human models, which is highly relevant for any preclinical pipeline. And to achieve this, the model should incorporate all BBB key components in order to improve physiological relevance. And in particular, the combination of all experimental systems, from plate-based screenings over mouse and human organ-on-chip models to in vivo studies and ultimately clinical trials, may provide a more comprehensive understanding of drug behavior and also BBB permeability. And therefore, our collaboration of Dynamic 42 and EPU hopefully helps to bridge the gap between early stage drug development and clinical trials. Thank you for your attention.
Thank you, Michelle. Thank you, Amélie, for your great presentation. Before we move into our panel discussion, I just want to quickly show you that we've got some training upcoming. So if you're interested, especially in cancer chip applications, we've got an advanced academy where you can learn how to integrate cancer and immune cells coming up in November here in Jena with us. Tom will be there, I'm pretty sure, so you can ask him lots of questions. So yeah, if you're interested to join, let us know and we can register you. There's still some places left. Okay, so yeah, we're moving into panel discussion now. Obviously, afterwards, there will be plenty of time for questions, so please keep them coming. I can already see some good ones. So I will start the panel round with a question directed to Jens. And I would like to know, like from a scientific and clinical perspective, what gaps do you see and what gaps do BB on chip models help close, and where do you see like the biggest opportunities for such models in preclinical workflows?
Yeah, thank you, Juliane. I think Michelle and Amélie have already introduced the high medical need we have in the clinic for glioblastoma patients. So there are really not many drugs available, and it's still a very difficult to treat disease. And currently, if you have a look from the research perspective, it's almost impossible to have in vitro models which can help you to understand whether a new drug which is under development is useful for patients with glioblastoma, because you have to model this blood-brain barrier. So it's not very helpful if you use a 2D cell culture and you put your drug on. It can probably work very well, but later, if you go into patients, you very often have a big disappointment. And here, this is really the invention of this model, of these lab-on-chip models, which can really help to improve this situation. And some of you may be already working in drug discovery, and you probably know the CACO2 model very well. So the CACO2 model is a model for the barrier in the gut and in the colon. And this is a very good model to understand whether drugs can really pass the barrier between the intestine. And so I hope and I think that this new blood-brain barrier model can really become as important as the CACO2 model in research, because it's really the first model that can help us to understand whether a certain compound can cross the blood-brain barrier, and then in the next step can really be active against glioblastoma cells first and then organoids, and can really support the development of new therapies for glioblastoma patients.
Thank you, Jens. And then to Tom, what do you think, like which features of the model are most critical for studying brain tumor biology and drug transport? Tom, I think you're mute, perhaps.
Sorry about that. So at the end, of course, the model is still being built, but there's a few features that we really like to establish first before we can really push this into the idea that it's really going to be predictive for the drug development process. Most important will be that the barrier is really functional. So of course, we have a lot of key factors that we first want to reach. Implementation of all three cell types, the barrier permeability really for selective drugs, so that drugs that should pass are passing and drugs that are not passing will not pass. And then, of course, once this is set up and we're really confident that we have a strong barrier that really only lets molecules pass that we know that should pass the brain barrier, blood-brain barrier. And then, of course, putting under that these different types of gliomas, glioblastomas, would be really interesting on the one hand to really just see efficacy of drugs, but on the other hand also see whether we can predict, for example, impact on the blood-brain barrier, where some of the tumors we know are impacting the permeability, whether we can also recapitulate this in the tumor. So maybe these two aspects would be really important. Replication of treatment efficacy on the one hand, and maybe also then replication of the impact and deterioration of the blood-brain barrier induced by tumors.
Thanks, Tom. And then maybe one more to Martin, like from a perspective of drug development projects, where are current models still lacking to answer key questions in glioblastoma research?
There are quite a few points that still need to be addressed, and we're talking here about a plethora of different models that we have. I mean, we have mouse models, zebrafish, we have the 2D culture, spheroids, tumoroids, organoids, and organ on chip. And there are questions that still need to be addressed. Things that have been named here, of course, most obviously is the barrier permeability, the selectivity of the barrier. We're talking about, as it was mentioned in the introduction, about blood-brain barrier, but also the blood-brain tumor barrier. A lot of the models that we're currently working with in vitro, but also when looking at animal models in vivo, are lacking the right human architecture we have in the human body, and sometimes the correct cell types we need. Another feature that we definitely need to be better represented in the preclinical stages is also the complete area of the immune system. So the immune microenvironment in such models plays a big role, and for instance in animals can be not captured fully due to species differences. And in simpler cell culture approaches, you just lack the techniques to really combine in a physiological way different cell types. And in the end, also a big question, I think, in the field still is standardization and decision thresholds. All these models we have at hand answer different questions, but we think that's why we also choose this approach and also working together, that a combination will be key to answer research questions and questions we have in the drug development better than we can now. And this is also a trend that is being discussed in recent reviews. So I think this is also something that is still lacking and needs to be done, combining different models.
Thank you, Martin. So, I would like to continue this panel. We've got so many good questions coming in from the audience, so I will move into those right now. We start with the first one, which I think I'd actually direct to Martin again. How robust are organ-on-chip systems, and do they require a really high quality or purity grade of reagents?
The robustness, I would say, is depending also on the cell types you're using. Some models run shorter times, some can run longer times. When you work with cell lines, robustness and reproducibility is higher than if you have to qualify primary cell batches and then also work with different batches. The charm here is, again, with a bit less robustness, more variability, you capture actually the situation in the human population better. So there's always a bit back and forth. But in the bottom line, these models can be as robust as other in vitro models. And you do not need special chemicals or cell culture material of special gradient, special quality for this. It works with the things you're used to work with in a cell culture lab.
Maybe one addition to that, because the blood-brain barrier certainly seems to be a little different here to other models. We do run here liver, gut, intestine models. there often we have a cell line-based model, which also performs really well, and then primary models, which are a little more expensive. So let's say more quality cells, but both can be used. And for these studies in the blood-brain barrier, we are finding that almost all cell lines which are out there are not performing like they should. So the blood-brain barrier per se is, of course, a very tough tissue to model. Many of the endothelial cells we were testing, but also others, don't behave really well in CHIP because they're not creating the 3D structure of the tissue that we are expecting. So maybe in some extenses, you need then higher quality cells to really be able to mimic that three-dimensional structure so that they don't just overgrow each other in the chip.
So the next one, I'd say, is directed to Amélie. Are you using primary cells from the mouse?
So mouse model is Michelle's work, so I would hand over the stage to you.
Yeah, I can answer that. We are working with different type of cells. The experiment results you saw was a immortalized endothelial mouse cell. However, I'm also working with primary endothelial mouse cells and also primary parasite and astrocytes, fresh isolated from the brains. If you mean the tumor cells, they are primary cells. So we isolate them fresh from the PDX tumors and then use them as primary cells.
Sure. I think the next one's to Amélie, but perhaps I'm wrong. Did you measure TIR in human endothelial settings?
Yes, we already did that. So there is actually the same biochip. We don't need to do any transfer. That has just added TIR electrodes to it. That doesn't change anything to the protocol. I've just seen that in the endothelial-only monolayer, the TIR values are not quite as reliable. So I'm just waiting on implementing I already have the parasites and astrocytes. And once I have stable co-culture, then I will have definite TIR values for that.
Then another one to you. Can you comment on differences of fusion rates between mouse and human settings?
I can also answer that. Yes, we can comment a little bit. So as you saw in the presentation, we can see differences. For example, that mouse endothelial cells can tolerate much higher perfusion rates compared to the human for now, because they are by themselves in a monolayer. And well, we think this could be because of the morphology and also the size of the cells. So the human cells were much bigger compared to the mouse cells, and maybe also in mouse brains, in mouse brain capillaries, the shear stress is much higher compared to in humans, because the vessels are much smaller. So this could be a reason, but we are still working on this and trying to figure out how to combine those or compare those.
Thanks, Michelle.
I would add here and also support this because we see this, or it is well known also from the literature, that we have lots of differences in flow rates between small species and larger animals, and especially in small species we have often significantly higher blood flow rates and pressure in the vessels. So I think biology could be here really also an explanation the origin of the cells, that they or answer if they endure more in a model or less in terms of shear forces.
Really, maybe last but not least, what we're really seeing also from other models is that the combination of multiple cell types really changes dramatically how the endothelial cells are reacting to the shear stress. So just adding a supportive layer on the bottom, which we know started with parasites, we already see a not much tighter, higher expression of tight junction markers on the endothelial cells. We haven't really put a lot of strain on them yet with shear stress, but we're getting there at the moment. So we're really positive about this kind of connecting then everything to astrocytes, and then eventually, hopefully, the human cells will also be able to address a lot higher shear stress, which of course is then one of the quality checks that we want to have for our blood-brain barrier.
And then to Amélie, are you testing static conditions only with the human BB-GBM chip?
So this is exactly building on the answer that Tom was just giving. We started with the endothelial cells first, and then encountered first issues that the human endothelial cells didn't withstand the perfusion as good as the mouse endothelial cells. That's why we chose a static condition for this experiment. But then again, as Tom said, when we will be adding parasites and astrocytes, we already expect that the biology is going to be the blood-brain barrier will definitely withstand higher perfusion rates. So we probably won't stay in the static condition.
Thank you, Amélie. And then one for Martin, I'd say. Are the chips available for purchase for the researchers? And is the system with active still prototype, or can it be shipped and set up in labs?
So the chips and perfusion system is available from us, can be purchased. So if you drop us a mail tomorrow, we can ship something next week.
Excellent. And then another question to, I don't know, Michelle or Amélie. How long can the current model run stably for?
Could go if you like. I don't care.
Go ahead.
Okay, sorry, my internet connection was not really good right now. We currently for about a week. So the usual experiments we plan is 3 days of static pre-culture, and then 3 more days of experiments, or 4 days. So I would say about a week.
Maybe also to comment on this from other models, of course, we can't predict exactly how the blood-brain barrier will behave. Generally, we are seeing this, that kind of a weak culture then of the fully established model is usually what a kind of sweet spot is. For some models, like in the cancer research, we could go a lot longer, but then often this would be without a vasculature, for example. So there would, if you just have tumors in chip, then you can go a lot longer. Generally, to be sure that these barriers are tight and intact, usually a week of culture, 2 weeks of culture. This is where usually the sweet spot is.
And then one question I direct to you, Jens. From patient biopsy to PDX model to bi-chip drug screening with results, how long do you think it would take?
This is a very good question. So if you really would first generate a PDX, this takes up to 3 months, and then go to the lab on chip. This will probably too long if you are thinking in studies like translational research to predict the patient outcome. In this case, one should consider to go directly from the patient material into an organoid and go into the chip. This would probably take up to 6 weeks, can be faster. But how we see this technology is not to predict the individual response for patients, but rather really support the development of new drugs to understand which drugs can pass the blood-brain barrier and really to work on glioblastoma. But one can also consider to use this model for addressing additional questions. So sometimes it's very important to protect the brain, the nerve cells in the brain, and some drugs are toxic to nerves, and you can here really see or evaluate whether a drug can pass the blood-brain barrier and can cause damage in the brain, and you can avoid the development of these type of drugs. And another very important research question, maybe also to look a little bit beyond this, what we have shown today is to use this model to evaluate the efficacy of new drugs against brain metastasis. You know that many cancer types for example, breast cancer or lung cancer, to metastasize into the brain, and there's still a high medical need for this type of disease. We can also hopefully address with this new model.
Yes. So this one I initially direct to Tom. Would this BB on chip model also be applicable to primary porcine brain capillary endothelial cells? Since the porcine brain is more similar to the human brain than the mouse brain, could this improve the translational relevance of the model, like animal to human translation?
So in the end, of course, you can.
Sorry.
We could certainly also combine this with other cell types from other animals. So there's no technical limit to this. Of course, within this collaboration, we really appreciate kind of having also the data then from the EPO side, who are doing mouse research. So in that case, for that specific project, of course, the idea of having really this human on chip blood-brain barrier, mouse blood-brain barrier with a human tumor, and then really the human tumor and the xenograft as really like kind of one line of evidence, then really kind of hopefully being translated into how it will perform in the clinic is the beauty of that specific model. Of course, if you yourself or somebody else also has then the porcine data available, this would be also, of course, very nice to study. And we are probably not going to do it anytime soon, but of course a good idea.
With the next one, but unsure, I initially directed to Jens. The chip appears to be a step up on mouse models, but has any work been done to compare it to gyrencephalic brain structure? Sorry, my speech is giving up on me.
Yeah, this is something which we are currently starting to evaluate, to really build up a much closer model to model the human brain structure. And this is what Michelle, I think, has shown in the last slide. So we are working on this, and we definitely also have to later to do the correlation between lab on chip and mouse autotopic mouse tumor models, and then, and last but not least, the patient results. But I'm quite sure that these models are of high translational relevance, and we will really receive very good results with these models.
And then one for Amélie, maybe also Michelle, we'll see. Do you also have a BB model for rat?
Not yet. we just do mouse and human right now, so
Yeah.
But as compared to the porcine brain, you can also do a rat blood-brain barrier. So you just need the right cells for that.
And then for Amélie, as glioblastoma induces important alterations in the vasculature, do you detect difference in vasculature and the presence and absence of glioblastoma cells?
So we don't have data on that yet, but that is definitely in the scope of this project and something we really want to look at, also maybe in different brain tumor entities. And then we can have different assay readouts for the BBB integrity, as we already presented, so permeability assays or tier values, looking at that, and then get an idea about the impact of the brain tumor on the blood-brain barrier.
And then another one to Michelle, I think. Question in one of the slides, viability was above 100%. Can you comment on that?
Yes, I think it was the human model viability. So we used the medium control without any treatment as a 100% viability line. kind of. So we were assuming that for our current model, this is the 100% viability, and then we were calculating up or down. And because in the human model, the viability in the control was, in this case, a little bit lower compared to the treatment, which is only depending on the glioblastoma cells in the bottom channel. So if the drugs did not pass through the barrier, they're kind of the same conditions in the bottom channel. So they're almost similar, and that's why just because we used the control as a 100% viability. Yeah.
Maybe a short addition from my side. It can happen always that under the treatment, some tumor cells change their behavior, and even the growth. Some drugs can even accelerate the growth of some tumor cells. This is if they are becoming resistant. Some drugs can really activate certain pathways. Tumor cells are highly mutated, and they can change their behavior and can sometimes even grow faster than without a certain treatment, unfortunately.
Yeah. Maybe also to add to this, and I fully agree with Jens there, we see this actually quite a bit actually in our tumor models, especially when drugs at a not optimal, suboptimal concentration are impacting the endothelial barrier, but not yet are really impacting the tumor underneath. So endothelial barrier is impacted, suddenly the tumor actually has more media to grow with. Often also, of course, in our systems, oxygen and nutrition is always coming through the vasculature. And now the tumor actually has more to live on than beforehand. And so these drugs are actually impacting the endothelial barrier, but by doing this and by not being potent enough to kill the tumor underneath, the tumor might actually be doing better. And maybe we are seeing something similar here, that we are at this kind of concentration where maybe the drug even is actively transported out, right? There's transporters that might here in this case really keep the concentration in the tumor department low. And then but the maybe a slightly higher accessibility to oxygen and nutrition from the vasculature channel might actually improve also the survival of the tumor cells underneath. So in this case, overrepresentation after drug treatment is sometimes even true.
Thanks. And then one more to Jens, I'd say. In brain tumor patients, edema due to impaired BBB can be a problem. This addressed by treatment with dexamethasone, PGP inducer, resulting in reconstructed stronger BBB. Can you capture these alterations, PGP inductions, in your models?
We have not done this yet, but it's a good idea, and maybe we can address this in the future here.
So maybe just...
Amélie, please go ahead.
Sorry. Yeah, to add on that, one of the readout assays we haven't shown here, but are already in establishment, are not only qualitative expression of proteins, also we were looking at PGP already in IF. We can also dissociate the cells and then do quantitative analysis, for example, of PGP expression then in flow cytometry, to then see these alterations, for example.
Yeah. And I just wanted to say I would also look positive into that experiment and hope to see some effects. What I can tell is that we see definitely effects of dexamethasone on barrier tightening in lung models.
Thanks, Martin. And then just to conclude this webinar, one last question to wrap this all up. So, Martin, what do you think? What will be needed for BB on chip platforms to become a standard in CRO workflows and for broader adoption in drug development in general?
For the models itself, I really would like to see, and I hope that we achieve this, to have a fully comprehensive model with all the relevant cell types and microenvironment that we want to have, that is needed to study this. And on the other hand, also more education about new technologies not just speaking about organ on chip for our own purpose, but in general, there's a lot going on that I think can be beneficial to get better and more predictive models in the end. Also, what I always like with in vitro, when it comes to then correlating this to in vivo situations, to combine it with computational modeling.
Yes, very good point. Jens, would you like to add anything to that last question?
No, I think I can fully agree what Martin has said. What I think what is necessary is really then to standardize a bit more the different cell types we need in the model. So this is currently a challenge. So we probably would need a blood brain endothelial cell line like the KECO2 cell line, so which can really be used in large scale. This is probably currently one of the most challenging things we have to address. But otherwise, I'm quite confident that this model will definitely have in the future a place in drug development and in better understanding how the blood-brain barrier can be overcome or can work.
Thank you so much, everyone. We'll conclude the webinar with this. If we haven't answered your question, we will definitely get back to you after this. And I will also send out the recording. So yeah, thank you everyone for joining us today, and we hope to see you again in the future. Goodbye.
Goodbye.
Bye.
Bye-bye.
Bye.